Welcome everyone, and thank you for joining us at the Fall 2020 GRASP seminar today. As a quick reminder, recording of this and previous seminars can be found in our YouTube channel and website. Now I would like to introduce our GRASP faculty host for today's seminar, Dr. Kostas Zinelidis, Ruth Yolom Stone Professor at the Computer Information Science Department within the School of Engineering at the University of Pennsylvania. Thank you.
Gabi, thank you for the introduction of the introduction. Welcome everybody to our virtual GRASP seminar. I'm very glad to introduce to you Jim G, our today's speaker. I know Jim since almost 25 years when I first visited to give a talk at the GRASP Lab. And I have really followed his work. Jim is probably one of the very first who did deformations for medical imaging. I mean, the paper, his paper from 1993 is really very well cited, and still many ideas from that time are coming even back now as trying to find diffeomorphisms to do 3D reconstruction. He's really widely cited. He's really, I mean, a pioneer on the medical deformable and the deformable models in the medical area, in medical imaging. He's a faculty in radiology with a secondary appointment in the computer information science. Jim is the director of the Penn Image Computing and Science Laboratory. He is the co-director of the Translational Biomedical Imaging Center. He received his bachelor in CS and electrical engineering at the University of Washington, as well as his master's. And then he continued his PhD at the GRASP Lab under the supervision of Ruzena Bajcsy. He's a distinguished investigator of the Academy for Radiology and Biomedical Imaging Research and a fellow of IMBE, SPIE, and ISMRM. Jim, I'm really so glad to have you today with us.
Great, thank you, Kostas. Let me share my screen. Can we see my screen?
Yes.
Yep. Okay. Fantastic. Well, thank you again, Kostas. I admit I feel a little, actually a lot sheepish to have this entire seminar to myself today when GRASP faculty had just 5 minutes last week to present, because my talk really is a continuation of last week's overview of research at GRASP, as I very much like to think of our lab as one of the GRASP outposts here at the medical school. So in the same spirit as last Friday's research overview, I'm going to showcase today the work of various members of our lab. Now, before I do so, I'd like to invite you to join me in pausing for a brief moment to reflect on and affirm our commitment to addressing racism, racial injustice, and social inequity. Uh-oh, it doesn't look like my screen is, oops, there we go. Well, thank you. And on a happier and much brighter note, let me start by asking, how many of you were able to attend the GRASP at 40th celebration almost exactly a year ago? Now, I'm sharing my screen, so I can't, well, it's virtual as well, so there's no opportunity for me to see hands up or actually if there's any responses on the chat line. So I'm just going to assume that hopefully many of you did make it. The celebration was just really an incredible event, I think, for old-timers like myself, Kostas included, and a lot of other folk. It was really a wonderful walk down memory lane. And I think it's fitting for me to elaborate today on the experience that I talked about during the brief remarks I gave at the celebration. I hope too that by doing so, all of you here will gain a better appreciation of and take pride in the seminal impact that GRASP has had in the domain of medical imaging and the lab's influential track record that continues to the present day. And of course, there's no surprise that Ruzena Bajcsy figures prominently in this narrative. We heard a wealth of testimony at the GRASP at 40 celebration, paying tribute to Ruzena's remarkable accomplishments, including the amazing fact that for all her achievements in robotics and computer vision, for which she's renowned, and that later earned her membership in the National Academy of Engineering, Ruzena was actually first elected to the National Academy of Medicine for the development of the first digitized anatomy atlas of the human brain and matching algorithms for computed tomography, magnetic resonance imaging, and positron emission tomography scans. Now, what's astonishing is that this landmark research on elastic matching and deformable atlases, which has become the foundation on which some of the most important work in the contemporary field of medical image analysis is built, this research was developed concurrently with a work on active perception, among other things. And I like to think this diversity and exchange of ideas is a hallmark of the work that takes place at the GRASP Lab, and a lasting legacy of Ruzena's principles and vision on how the best science can be facilitated. Now, the developments cited by the National Academy of Medicine were preceded by another set of events at Penn that would revolutionize the imaging of human brain function. It was at the Cerebrovascular Research Center at Penn that positron emission tomography, or PET's potential for quantifying a regional brain function, such as blood flow and volume, was first established. The radio tracer fluorine labeled deoxyglucose, FDG, developed by Martin Rajvich and Abbas Alavi and coworkers, has been hailed as the quote-unquote molecule of the 20th century for its unparalleled and unique impact on the evolution of the field of nuclear medicine. Now, the emergence of FDG PET prompted the need for new analysis methods and resulted in the collaboration with Ruzena that gave rise to the work on elastic matching and atlases. In particular, a method was needed to anatomically localize the activation image in the PET scan in order to identify the cortical networks subserving the cognitive processes under examination. In other words, they had these incredible pictures, as you see here, for the first time of brain metabolic activity as it corresponds to regional glucose uptake, but they needed to know where exactly in the brain this activity was taking place in order to fully make sense of what they were seeing and measuring. It's no different than, say, having a heat map of new coronavirus cases in the past 14 days. The image or data becomes much more useful when we have more precise information about where exactly the hotspots are located. What state, which city, which county, for example. Now, to address this problem, Ruzena, working with her student AIMBE, leveraged the fact that, to a certain level of detail, the anatomy of normal individuals can be thought of as quantitative variations on a common underlying qualitative plan. In other words, we all effectively share a common anatomical or bodily blueprint. Now, this being a virtual seminar, I can't verify just by looking around at all of you who are attending this talk online actually have two eyes, a nose, a rough symmetry midplane, and so on. But I'm going to assume that there's a pretty good chance, a pretty high likelihood that that's actually the case. Now, at the same time, there are obviously differences among us, but we can think of these differences as representing variations on this shared blueprint or this template. So my nose may be a little smaller or bigger than yours. My face may be rounder, and so on. Ruzena proposed that a natural way of using this knowledge about human anatomy, the face in this particular example here, is to imagine that a given individual's anatomy or face is a warped or deformed version of some template or reference anatomy. And if this warping function could be calculated, anatomical features or structures can be identified just once in the reference anatomy and then automatically identified in an individual through the warping function. And things can get even more interesting because we can annotate our reference anatomies with additional kinds of information as shown here. In other words, what Ruzena and Haim had conceived was the first ever digitized atlas of anatomy. The fundamental problem then in this approach to localizing anatomy is to first define the class of admissible warping functions or transformations, which have to be broad enough to enable the reference anatomy to fit all subject anatomies. And a second, to develop efficient automated algorithms for the calculation of the appropriate transformation. Now, in developing what became to be known as elastic matching, Ruzena and Haim sought out a method that would optimally balance the similarity induced between the registered image pair with the amount of deformation to the transformed image required to achieve the registration result. This reference to the kinematics of the solution was made explicit in the formulation of the problem of matching two images as one akin to the physical process of manipulating and applying forces to an elastic version of the object depicted in one of the images so that its deformed configuration resembles the target object in the second image. The appropriate forces or loads are derived from a potential function, the similarity term in image registration parlance, that provides at each point of the object under deformation a measure of the quote-unquote similarity or correspondence between that point and any of the points in the target object. And the first object is deformed in this way until an equilibrium configuration is reached, or equivalently, until the total potential energy of the system is at a local minimum. Now, by choosing the elastic strain energy as the measure of deformation, the overall cost or objective function for the image registration problem becomes the total potential energy. And the elastostatic configurations, which locally minimize this cost, represent solutions to the corresponding elastic matching problem. And these groundbreaking ideas developed more than 30 years ago brought about a paradigm shift in medical image analysis from slice by slice, two-dimensional processing to fully three-dimensional analysis. A paradigm shift that was a precursor to active contour and physics-based deformable models that later became pervasive in the field. And here are those ideas implemented for the first time in code and applied to a very early computed tomography study of the brain. And a more contemporary version of the same application, this time using magnetic resonance imaging or MRI, which has become the preeminent modality for in vivo imaging of soft tissue. The results shown here are from our lab, represent the efforts of a good number of people, in particular, the individuals pictured here, Brian Avance, Gary Zhang, Phil Cook, Jeff Duda, and Paul Yushkevich, whose work you'll be hearing more about in a little bit. These developments in atlas-based segmentation or parcellation of images into anatomic regions of interest allows for an automated approach to the regional quantitation of image signal values, as well as morphology, that remains perhaps the most popular method for hypothesis-driven analysis of medical imaging data. And shown here is a typical example of this sort of region of interest-based study, where in this instance, differences in brain region size were not only contrasted between patients with schizophrenia and healthy individuals, but further correlated with age, which allowed us to test a theory that's been proposed of disordered brain maturation in this disease that, if true, would provide new opportunities for treatment approaches and strategies. And this work, among others, was led by Abraham Dubb, who was one of our earliest clinical fellows at the lab and has long since returned to the clinic and to taking care of patients. Now, once the capability of taking two or more image volumes and registering their fine details was established, and an incredible tool was unleashed. Data from different modalities could be fused or integrated to facilitate interpretation. As an example here, where ventilation imagery from hyperpolarized helium MRI is registered with the lung CT of the same subject. And what's more, we show as well the specific airway branch segmented from the CT image that's associated with the particular ventilation defect apparent here in the patient's helium-3 image. And that's our former fellow and longtime collaborator, Nick Tustison, who's pictured here and remains our lead on this and much of our other lung imaging work. Now, the ability to register images also allows changes or differences in morphology to be detected and quantified. And this is because the registration or correspondence transformation that connects the images directly tells us in quantitative form how the object structure changes or is different between its representation in the different images. And you can see this in the paths that points within the circle shown here travel along as they are transformed into the corresponding positions in the square. Note how these paths or displacement vectors point outwards, indicating that the smaller circle must expand in order to fill out into the larger size of the square shape. You can see as well the refinements of these paths along the boundaries, which quantitatively inform about the differences in the shape between the circle and the square. And what's shown on the animation on the right-hand side is the analogous transformation between a chimpanzee and human brain. However, if I were to draw out the paths that points take for this transformation, they'd actually be curved instead of following a straight line. They're really flows. If you squint your eyes a little bit and look carefully enough, you might be able to see this effect within the frontal cortex as it's transformed from its chimp appearance into its human configuration. Now, this curvedness is a feature of the diffeomorphic property of the transformation solutions we now compute, a property that derives from exactly implementing Ruzena and Haim's original continuum assumption. What's exciting about this development is that, among other things, it's led to a rigorous formulation of a distance or length metric defined over the transformations or diffeomorphisms. And what this translates to in practice is the availability of a quote-unquote yardstick or ruler for anatomy that allows one to properly measure the distance between two instances of the same anatomy. And this is hugely important, right? The existence of this proper metric allows us now to compute statistics on organ anatomy and perform morphometry, for instance, to calculate the average brain configuration of a group of individuals, as shown here in the work that was carried out with a former mentee, Yanel Lebricker. We're able to compute principal components and so on and so forth. This is really at the very cutting edge of computational anatomy and is a very active area of research as well as application development. So for example, the chimp and human brain transformation that I showed in the earlier slide was part of a collaboration with Tom Schoenemann, a physical anthropologist formerly at Penn and now at Indiana, who remains interested in questions surrounding human brain evolution that could potentially be informed by knowledge of exactly how human brains differ from those of our closest ancestors. So to compare across species, their average brain configurations are calculated and used to compare shape distributions between the species. So in particular, a brain region of special interest is the prefrontal cortex, involved in planning complex behaviors. And our analysis confirmed that this region occupies a proportionately larger part of the brain of Homo sapiens than it does in pan troglodytes or chimpanzees. And the identical methodology can be used to parameterize morphological variations across human health and disease. As for example, in a study here of frontotemporal lobar degeneration and of Alzheimer's disease, where because these dementias can be confused clinically, there's a good deal of interest in establishing new neuroimaging biomarkers that can help in distinguishing between these two disorders. Another way to support disease differentiation is to examine the time course of disease progression. And here again, we highlight the critical role that image registration plays in medical image analysis. Namely, its use in registering a sequence of longitudinally acquired images to capture an estimate of the annual atrophy, in this instance, in a group of frontotemporal dementia patients where loss of gray matter is evident in the frontal and temporal lobes, shown in a red-yellow scale here with relative sparing of the occipital lobes. That's a hallmark of this dementia. The same annualized atrophy result is shown animated here on the right with respect to the average brain configuration derived from the frontotemporal dementia patients. And what's visualized is the average amount of gray matter loss in just over a year in our group of patients. Well, hopefully it's not too difficult for you to see how these kinds of longitudinal studies can have an important role in not only improving our understanding and diagnosis of disease, but also in their treatment in terms of monitoring and determining a therapeutic efficacy. And this work is just a small part of a decade-long partnership our lab has had with Murray Grossman, who directs the Penn Frontotemporal Degeneration Center at the medical school. Now, a different sort of serial imaging study is shown here in work by Tessa Cook, a former PhD trainee at the lab and now radiology faculty at Penn. In this situation, the moving lung presents a different kind of anatomical variation that can nevertheless be characterized in the same way that we've modeled, for example, intersubject as well as interspecies variations. And this illustrative study developed by our collaborators at the University of Virginia really elegantly demonstrates the potential of measured motion patterns and the corresponding mechanics to assess detection of localized pathological processes, as well as to evaluate therapeutic intervention, as in the specific example here, where a patient with idiopathic pulmonary fibrosis has undergone right lung transplantation. I especially like these pulmonary deformation studies because they additionally showcase the finite element method that was used in certain applications to compute the registration solutions. And this has opened the registration framework in a natural way to applications in biomedical modeling, such as the pulmonary studies we've just seen. And this direction of work was one of the fruit of another longstanding collaboration, this one with David Hayner, a longtime mentor and colleague who's a mathematician and a radiologist at the University of Washington. Well, summarizing the developments which have made possible all of these studies and many more, they're continued mechanical deformations in variational formulations of registration, digital anatomy atlases, multi-resolution strategies, and optimized image representations for both similarity calculation and warping. Both of these I actually didn't have time to cover, but were major contributions of the landmark multi-resolution elastic matching paper that Stana Kovacic, now retired professor at Ljubljana, co-authored while a student of Ruzena's. Finite element implementations, as you've just heard, and Bayesian formulations, and its introduction of probabilistic similarity measures and empirical priors on registration mappings that, again, in the interest of time, I had to skip over. Now, these are some of the foundational elements over more than 2 decades of inspired and visionary work that Ruzena carried out in the field. All this work originated under her watch. This is really a track record that remains unmatched among her peers. And I'm terribly proud to have had the good fortune and privilege of working on many of these developments with Ruzena, who was my thesis advisor, as you know, and even more proud of the subsequent and continuing innovations introduced by my own trainees and members of our group, including even more advanced formulations that have become standard in the field. Now, I mentioned earlier, for example, that our correspondence mappings are now optimized over the diffeomorphisms. These are just a set of differentiable maps with differentiable inverse, in other words, the topology of the domain. In this case, the image that's being transformed or normalized is maintained or preserved by the transformation mapping. These diffeomorphic maps are generated by integrating a time-dependent vector field. This is just a velocity field effectively over time, starting from X, the identity configuration, And the idea here is that by ensuring that the velocity field is smooth, the displacement mappings we get by integrating the velocity field will themselves be differentially smooth. In other words, there'll be diffeomorphisms. There are the standard constraints on the optimization problem. The first is that at time 1, the diffeomorphism should map image I, shown on the left here, into the space of image J, shown on the far right. The second constraint is that the length of phi, the mapping, should be as small as possible. And this length is described by the integral over time of the velocity field size. And so by solving this problem with these constraints, in other words, the registration problem, we can smoothly map image I into the space of J, as shown here. And here we see an animation of a diffeomorphic map in this example between the chimp and human brain that we saw earlier. Each frame in a time lapse of the movie corresponds to a point along the diffeomorphic map. And we can get some initial insight into the algorithm by taking a look at how it discretized the problem. As we saw in the preceding slide, the dense in space and time velocity is sampled at a finite set of time points. And looking at the integral, we can see that the initial value of the diffeomorphism is just the identity, phi x times 0 equals x itself. And to update the value over time, we simply traverse the velocity field, accumulating the deformation in small time steps until time 1 is reached. Now, as one might imagine, this looks locally at a voxel like a curve integral. And indeed, this is exactly what it is, except for that it's spatially dense in a continuum instead of at a single point. So for that reason, a diffeomorphism locally over time traces out a curve, which is defined uniquely by its initial value, the identity, together with the tangents of the curve described by the velocity field, Vxt. And so to optimize the diffeomorphism, we basically just manipulate the values of the velocity field. It's important to note that this is a very distinct concept from just simply applying a series of incremental deformations to an image, something that's conventionally done to mimic large deformation transformations. Now, the trouble with this approximation is that it doesn't guarantee that domain topology of the warped to transformed image is preserved. It also doesn't ensure that a proper inverse mapping will be available, and we're going to see very shortly why this is so important. Now, there is, however, quite a serious shortcoming of the approach that I've just presented, and in fact, with all standard registration methods in general. And that's the fact that the formulation and the viewpoint it takes are fundamentally asymmetric. The path from image I to J, and the opposite or inverse path from image J to I are usually not the same. What that means is that the metric distance between our endpoints, in other words, the total deformation summed in time, actually depends on which image we choose as the reference point. In other words, the direction in which the registration is performed actually makes a difference. And this bias is clearly a problem for the kinds of quantitative applications we're interested in for any of our research or clinical imaging studies. Now, fortunately, there is a way to fix this problem and to see this. The idea that was developed as part of the beautiful thesis work of Brian Avance, whose picture is shown here along with Charlie Epstein at the math department, who also contributed to this work. The idea is, again, to generate the mapping phi in the same way as before as the integration of velocity field. Except for what we do now is we move the endpoint condition to the middle of the path by explicitly representing the component of the diffeomorphic map that operates on image J. Now, what this means is that the geodesics, or the shortest diffeomorphisms connecting the two images, are now required to be the same. And this now gives us a set of symmetric Euler-Lagrange equations that depends on gradients of both images I and J, as opposed to just image I in the original formulation, which is the root cause of the asymmetry that is the basis of the bias in standard registration. Now, the second important numerical difference is that the optimization can also be carried out symmetrically now. So we not only have a symmetric formulation, but the optimization can also take advantage of this. And the way we do this is we discretize in time the velocity field as we did before, except we then update the velocity field symmetrically from both endpoints with initial phi values, and then generate new phi values by integrating as before the velocity. And this process is just repeated until convergence. Now, I've been told sometime that the method, as I've just presented it, can be a little bit opaque. So if that's indeed the case for any of you out there, please don't be concerned. The basic algorithm is actually really quite easy to appreciate when we take a look at the symmetric normalization in action. So in particular, let's consider as shown here the sad face on the far left is image I, and the happy one in the middle is image J. And I'm using smiley faces here only because it's easier to show the effect of the algorithm with these cartoon faces than say with brain images. So note where conventionally one image is typically deformed into the other. Both images now deform into the mean configuration at time equal to 1/2. And a total symmetric normalization or path between the two images is obtained by composing the two diffeomorphisms shown here. So rather than going from sad to happy I to J, or happy to sad J to I, both cartoons first become sappy. That's a sad attempt at humor. And so to go from sad to happy, for example, you start with the depressed state and you flow into sappiness. And from there, you take the inverse mapping from sappy to happy. And the path from I to J is now identical to the path from J to I. And what's even better is that this symmetric registration not only gives us unbiased results, critical to quantitative studies, but also results in significantly improved performance because all of the images now contribute to the calculation. As we saw earlier when the gradients were shown to be taken with respect to both images in the pairwise registration problem. And this performance gain has been essential in enabling ever more precise and accurate results, currently top ranked in the field. It's also made possible wholly new applications as well. And an important one I'll highlight here is how the symmetric normalization approach naturally generalizes from pairs of images to define a symmetric population study over arbitrary numbers of images. And an easy way to see this is to note that the pairwise symmetric normalization algorithm effectively solves the two mean problem. We saw how we actually solve for the mean when we go from I to J and J to I, and then transition with the mean configuration to actually get from I to J or J to I. Well, a population study simply corresponds to solution of an arbitrary n-mean problem, n being the number of subjects in the group analysis. And this is exactly what the generalization provides. It finds an optimal reference coordinate system or template I bar given directly by the n-mean configuration, which by definition minimizes the total length of the set of diffeomorphisms phi operating on our subject images ji. And these diffeomorphisms are exactly the population normalizing transformations of interest to us in a group study. Here's another extension that forms some of the critical groundwork that gave rise to a major research direction in the lab that you'll hear more about shortly. What's shown here is the three-dimensional reconstruction of a stack of two-dimensional missile sections into what ultimately became one of the original versions of the Allen mouse brain atlas. And without going into detail, let me just say that a method effectively estimates a smooth approximation to the volume in the analogous way that one smoothly approximates a curve through a given set of points. Here, the approximated quote-unquote value is a deformed version of a given two-dimensional section I that minimizes the length of the mappings to its neighboring sections. In other words, the maps from section to section parameterize the volume, and the length of this parameterization, or the total deformation, measures how much discontinuity there is between sections. And so minimizing parameterization's length makes the section-to-section deformation smoother, and this in turn produces a more smooth change in anatomical shape within the final reconstructed volume. And with that, I'm going to wrap up my speaking part of this presentation and turn over the remainder of the talk to recordings made by some of the other faculty members of the lab that spotlight the really exciting work they're leading at PICSL. First up is Alison Pouch, who is newly appointed this year as joint faculty in radiology and bioengineering. Alison trained at Penn as an HHMI-NIBIB Interfaces Scholar, which is a program that I direct and help develop between the engineering and medical schools, where engineering students effectively join the medical school and train with medical school students for their first 18 months at Penn. The idea being to better promote and advance clinically oriented work by not only embedding our students within the medical environment, but also thoroughly immersing them in a clinical culture. And Alison has just been a perfect poster child of the program as you're about to see.
Hello, my name is Alison Pouch. I'm an assistant professor of radiology in the Penn Image Computing and Science Lab. And today I'm presenting our work in image analytics for cardiac and vascular surgery. The focus of this presentation is how we can use automated image analytics to help standardize new complex surgeries like bicuspid aortic valve repair. First, I'd like to give a brief overview of my research. My expertise is in 3D and 4D ultrasound segmentation and methods for anatomical shape modeling. Much of my research is focused on interactive visualization of heart valves for preoperative surgical planning. So anytime a patient comes into Penn for a heart valve surgery, they have an intraoperative transesophageal imaging exam done, and those images are used to help with surgical planning. Our goal is to develop segmentation and shape modeling algorithms so that we can represent the shape of the heart valve in those images so that the surgeon can interactively visualize the heart valve before the operation and make any quantitative measurements that might be useful for risk stratification for surgery. An example is shown on the right. It's a reconstruction of mitral valve motion that was extracted from one of these transesophageal echo exams. Now, when this work began, we started by segmenting and modeling the shape of the mitral valve with applications to mitral valve repair surgery. Now, mitral valve repair is a procedure that's been under development for almost 40 years now. And when I started this work back in 2009, mitral valve repair with annuloplasty was pretty aggressively recommended for treatment of ischemic mitral regurgitation. Over the last decade, however, we've witnessed high failure rates reported in patients with valve repair for ischemic mitral regurgitation. For example, up to 60% of patients experience moderate to severe disease recurrence within 2 years after the operation. Now, there are a number of reasons why this might be the case, but unlike 40 years ago, we're now able to reconstruct models of the mitral valve before the procedure and after the surgery. And as you can see here, it's common after mitral valve repair for the mitral valve not to look like a physiologically normal valve. For example, there's flattening of the mitral annulus and tethering of the mitral leaflets. And it might be the case that this excessive tethering might actually exacerbate stresses on the mitral leaflets, which may not be conducive to a good repair outcome. So over the years, we've been trying to identify preoperative morphological features of the mitral valve that might be predictive of disease recurrence after repair. And some features, like the leaflet tethering angle shown here, we believe might be potentially predictive of a poor outcome for mitral valve repair. And patients with these preoperative features might be better suited for valve replacement. So along these lines, we're interested in using advanced image analytics to help reduce the trial and error involved in developing new surgical repair strategies. So not only improving outcomes for mitral valve repair, but also for newer complex techniques like repair of bicuspid aortic valves. So the bicuspid aortic valve, or BAV, is a congenital defect where the aortic valve has 2 cusps rather than 3. In patients with BAV regurgitation, the conventional treatment is valve replacement. However, these patients tend to present when they're young, between 20 to 50 years old, so replacement really isn't an ideal treatment, and a durable BAV repair is certainly preferable. Unfortunately, however, the approach to BAV repair planning and the repair itself are not quite standardized. To give an example, the metrics for determining BAV repair candidacy aren't founded on strong evidence. So cusp height, to give an example, is a measurement that's used to determine whether a BAV is repairable. And this measurement isn't typically made on imaging, it's usually made on the intraoperative exam when the heart is stopped and the valve is exposed. And here you see it being measured with a ruler just before the procedure. Now, the thresholds on cusp height that are used to determine if the BAV is repairable is based on data from tricuspid aortic valves, and that data may not and probably is not translatable to bicuspid aortic valves. So this is just one example of why this planning for BAV repair is not standardized and still remains somewhat controversial. So how can we use automated image analytics to help standardize BAV repair surgery? Well, the first step is we can develop automated 3D and 4D comprehensive preoperative BAV assessment tools, very much like the tools that we developed to analyze the mitral valve with applications to mitral valve repair surgery. And the idea is that the surgeon would be able to interactively visualize the BAV before the surgery and make any quantitative measurements such as cusp height and new more comprehensive measurements like cusp surface areas to help with risk stratification. Those types of tools are not currently available for congenital heart disease. And secondly, we want to go back to the basics. Currently, there's no data set that shows us what physiologically normal or competent BAVs look like. We don't know the anatomical and dynamic features that define valve competence in these patients. And the goal of valve repair is really to take a diseased valve and restore it to a normal functioning geometry. And without knowing what that geometry is, it's very difficult to standardize the repair. So our second goal here is to acquire images that capture BAV competence and use that image data along with our automated tools as evidence to optimize BAV repair strategies. So as a starting point towards these goals, we developed a 4D semi-automated segmentation tool for analyzing the aortic root and transesophageal data. Now we started first with the aortic root because that's the simplest part of the geometry of the aortic valve apparatus. It's the cylindrical part of the outflow tract that you see here in yellow. So our pipeline takes in transesophageal echo data that we acquire in the operating room. We perform a segmentation at each phase of the cardiac cycle and output a series of meshes that show root dynamics over the cardiac cycle. And a video of that motion in a BAV patient is shown here on the right. Now conventionally, the aortic root geometry is assessed at a single time point in the cardiac cycle using diameter measurements of the aortic root. And we wanted to take this analysis to the next level and make cross-sectional area measurements of the aortic root at multiple levels of the aortic outflow tract from the ventriculo-aortic junction to the sinotubular junction, and to study area change as well as root volume change over the cardiac cycle. In a preliminary study, we analyzed root dynamics in a collection of BAV roots from patients with moderate to severe regurgitation, and we compared it to root dynamics in patients with competent bicuspid aortic valves and physiologically normal tricuspid aortic valves. These are a few of our 3D reconstructions of the aortic roots of our BAV patients. And you can see they vary considerably in morphology and size, as is expected with BAV patients since they often develop aortic aneurysms. The primary finding of this study is that BAVs with moderate to severe regurgitation undergo larger normalized volume change during the cardiac cycle than aortic roots of competent valves. And this data is showed here in this graph. So this is a finding that that's very interesting, and we're further exploring it for its implications on surgical interventions. It's also interesting in terms of understanding why certain BAV patients develop aneurysms, and also perhaps understanding rupture risk in those patients with aneurysms. In addition to analyzing the aortic root, we most certainly want to understand the morphology and dynamics of the BAV cusps. And this is a much more complex problem. So at this stage, we're currently atlas building. We're developing a repository of expert traced models of the BAV cusps. And we're capturing details like the raphe, which is the area of cusp fusion in BAVs. And the goals of developing these, this reference atlas set and the segmentation tools is, again, visualization of the mechanisms of regurgitation in these patients before the surgery is performed, and development of preoperative metrics for valve repair candidacy. And again, those metrics might be related to cusp height, cusp surface area, annular geometry, and other features of valve and root dynamics. We're also working towards a multimodal imaging approach. We don't want to just use echo for preoperative planning in the operating room. We can also combine it with other data, for example, CT, to help augment preoperative surgical planning. I'd like to give an example of how we can apply these automated image analytics in addition to just visualization and measurement extraction. We collaborate with mechanical engineers, particularly the SACS research group at UT Austin, to use these image-derived models as input to simulation. And the long-term goal is that we would take an image, a preoperative image of a BAV patient, construct the geometry of their BAV, and run simulations of different repair procedures to see what the outcomes of different surgical approaches would be. So the basic idea is that we would be performing a valve repair or multiple valve repairs before a surgery is ever performed. So you can optimize the surgical intervention before the patient ever enters the operating room. So as a step towards that goal, we show some data here where we have the aortic cusp at one phase of the cardiac cycle, and we simulate valve closure. We can also estimate tissue mechanic parameters like stretch patterns of the cusps and strains in tricuspid aortic valves, as well as BAVs with different leaflet fusion patterns. I'd like to give another example of how we can integrate automated image analysis with tissue mechanics to help optimize interventions. And this is in the vascular research domain, a collaboration with the Jackson Vascular Research Group, including Jordan Stoker, Kevin Edinger, and Benjamin Jackson. And in this work, we're exploring improving rupture risk prediction and also optimizing timing of interventions for abdominal aortic aneurysms. And the idea is that we're segmenting abdominal aortic aneurysms longitudinally in CT image data to produce locally resolved measurements like local growth of the aneurysm, which you see here in color. Now, conventionally, timing of surgery in these patients is dependent on a maximum diameter measurement in CT data, and also the rate of growth of the aneurysm. Interestingly, what we find in this study is that when you have locally resolved tissue growth measurements, we can see that the area with highest growth is actually not the area where they would be measuring the maximal diameter. So this is an example where more advanced automated image analytics might help improve rupture risk prediction in abdominal aortic aneurysm cases. And finally, I'd like to conclude with a summary of our research program goals. Our overall goal is to develop computational image analysis and visualization methods to guide surgical treatment planning, as in the example of mitral valve repair surgery. Also to standardize new complex procedures like bicuspid aortic valve and other adult congenital heart defect surgeries. And also transition many of these to minimally invasive or transcatheter approaches. We also want to engage in team science initiatives to integrate image analytics with tissue mechanics analysis, as I described with our simulation research. We can also use automated image analytics to help inform medical device design. And another idea that I didn't have a chance to touch on in this presentation is that we're investigating non-traditional methods of interactive image visualization, such as immersive interaction with echo data using virtual reality. I'd like to thank our funding sources and our collaborating research groups, and thank you very much for your attention.
So Alison's work is just amazing and at the absolute cutting edge. At the same time, you heard in her work echoes of ideas and concepts that originated a decade, or actually even 2 decades ago at GRASP. Next up, we're going to hear from Paul Yushkovich, who also trained at the lab and is now radiology faculty.
Hello, my name is Paul Yushkovich, and I'm a professor of radiology at the University of Pennsylvania. My work is in the field of computational image analysis, and lately has been particularly focused on postmortem imaging as a way to derive better biomarkers for Alzheimer's disease and related neurodegenerative disorders. Alzheimer's disease ravages the brain, but it does so in a non-uniform fashion. The earliest manifestations of the disease in the earliest neurofibrillary tau tangle pathology emerge in the medial temporal lobe, and even within the medial temporal lobe, they follow a well-specified progression. The earliest tau tangles occur in the transentorhinal region, and they spread through the entorhinal cortex to the hippocampus and elsewhere in the medial temporal lobe in the brain. This pattern of spread has suggested the use of targeted imaging biomarkers as a way of detecting and tracking the progression of early Alzheimer's disease. In fact, the hippocampus has been used as a biomarker for Alzheimer's disease for more than 3 decades. But recent advances in imaging, such as the use of high-resolution T2-weighted sequences at 3T and more recently at 7T, has allowed us not only to measure a large structure like the hippocampus, but to also measure changes in much smaller and more targeted regions, hippocampal subfields, as well as the extrahippocampal structures, such as Brodmann area 35 entorhinal cortex, which encompass among them the transentorhinal region. In our computational work, we have developed automated techniques for detection of these subregions and for measuring their volume and their thickness. We've been using them in a number of studies looking at relationship between these markers and Alzheimer's disease, relationship between these markers and in vivo markers of tau pathology, which underlies Alzheimer's, etc. As a matter of fact, over the last 5 to 10 years, we've published a number of papers linking subregional biomarkers in the medial temporal lobe to both disease progression, cognition, and tau PET. This table here shows some of these papers showing that we're looking at different imaging modalities, T1 and T2, cross-sectional and longitudinal studies, as well as in different cohorts, including the ABC cohort from the Penn Memory Center and national studies like ADNI. The consistent findings throughout of these have been the involvement of this Brodmann area 35, again, which includes as part of it the transentorhinal region, throughout a lot of these cross-sectional and longitudinal studies. So for example, the picture I highlighted here is an in vivo map looking at a correlation between tau PET uptake in the entire MTL and regional thickness throughout the medial temporal lobe. And what we see are the clusters of significant correlation between these two measures, encompassing the transentorhinal region in the Brodmann area 35, as well as parts of the entorhinal cortex and parts of the subiculum of the hippocampus. And I'll come back to this figure when I highlight some of our in vivo work, ex vivo work. So far I've talked about in vivo imaging. Why do we want to look at the medial temporal lobe using ex vivo MRI and histological imaging? And there are multiple reasons for this. In terms of structural imaging, when we look at these small subregions of the medial temporal lobe in vivo at relatively low resolution compared to the size of these structures, we're greatly confounded by large extent of anatomical variability. So in fact, the medial temporal lobe varies from subject to subject, from individual to individual, based on the depth of the collateral sulcus. And the depth of the collateral sulcus greatly affects the location of the transentorhinal region, as you can see on the panel on the right. So does the branching of the collateral sulcus. And in order to better detect and measure these important structures using in vivo imaging, we need to have a better handle on this anatomical variability. Some kind of a probabilistic atlas that describes the variability and describes the relationship between the locations of regions involved in early AD and the macroscopic anatomy of the MTL. Having this information, we can improve and validate our automatic segmentation techniques for in vivo imaging. And at a more global scale, we can validate findings from in vivo research using ex vivo imaging. Now, looking beyond just understanding what we're seeing on structural MRI biomarkers, we want to understand and quantify pathology in Alzheimer's disease. In particular, this classical study of Brack and Brack that described the progression of tau pathology through the MTL was performed in two-dimensional histology slides, and it does not describe the spread of pathology in 3D. And in the last 30 years since Brack's work has been published, there really hasn't been a good quantitative characterization of tau pathology in Alzheimer's disease in 3 dimensions and in a quantitative way. More importantly, we want to understand how pathology relates to neural loss, which in fact is what causes symptoms in Alzheimer's disease. And most interestingly, if we can accomplish this, we would like to use pathology quantification as a way of identifying hotspots for in vivo biomarkers. In other words, finding areas of the medial temporal lobe where macroscopic changes in thickness that we can detect using in vivo MRI are specifically indicative of disease progression in early AD. And this is crucially important because Alzheimer's disease is a heterogeneous disease. In fact, most people with Alzheimer's disease have comorbid conditions, including vascular dementia, as well as a number of other non-AD proteinopathies, such as TDP-43 proteinopathy and alpha-synuclein proteinopathy. And if we can identify hotspots that are specifically linked to AD pathology and not these other proteinopathies, we can develop better, more targeted biomarkers, both for, again, detecting disease, but most importantly for tracking progression and for supporting clinical trials of disease. So in order to answer these questions, we have established an ex vivo imaging pipeline in our group. It's done in collaboration with the Center for Neurodegenerative Disease Research, CNDR, and with the Penn Memory Center. When CNDR receives a brain donation in one of the individuals involved in their Alzheimer's Disease Center studies, we obtain one hemisphere from the brain. This is part of the Alzheimer's Disease Center protocol. We fix it in formalin for 4 weeks, and then we scan the entire hemisphere at 7 Tesla overnight. This gives us a high-resolution scan of the entire hemisphere, which is part of the service that the ADC provides, allowing these hemispheres to be banked and studied in relation to a multitude of other variables that are collected in these individuals, both while they're alive and ex vivo after autopsy. Afterwards, we extract the MTL region, the medial temporal lobe region, using a custom 3D printed mold. This is something that we have been innovating in our group, allowing precision extraction of the region with minimal errors. I will show a slide on that in a second. We then scan the medial temporal lobe region at even higher resolution on 9.4 Tesla animal scanner. We also rescan it at 7 Tesla to account for some gradient distortions that you unfortunately have to deal with on animal scanners. And we again 3D print a custom mold for guiding histology. We then perform histological blocking, sectioning, and staining. And then the computational stuff comes in when we reconstruct serial histology from each MTL with the MRI so that both are in the same space, and then register these images to a population-specific template. In other words, build a probabilistic atlas of the MTL that captures variability in the population. And finally, this series of registrations and allows us to perform analysis. So I will highlight a few of these steps that I find particularly interesting in our work. So one of these is a project that has been performed by a visiting student and intern from the university from a university in Lyon, France. She's only been with us for a few months, and she has developed this excellent program where with a few strokes of the mouse, one labels the MTL in a ex vivo MRI scan of the hemisphere. And then the program takes over and automatically builds a custom mold with cutting guides that allow a technician to cut the brain in a way that preserves the MTL, as well as preserving other parts of the brain that might be needed for other research projects, in particular frontal regions that are being used by our colleagues in the Frontotemporal Dementia Center. So this mold, first there's a hemisphere size mold, and then there's a second mold that allows trimming so that we can get a smallest possible piece of tissue that includes all of the medial temporal lobe substructures that we care about and fits into our 9.4 Tesla imaging coil. Here's an example of this specimen in the holder. And these are the examples of 9.4 Tesla images that we've collected. We have now collected over 50 scans of MTL specimens through various stages of Alzheimer's disease, as well as frontotemporal and other dementias. We image at 200 micron resolution. And on the bottom, you can just appreciate the great range of variability in these scans and just overall anatomical variability. So taking these scans and putting them into some kind of a template is by no means trivial given this variability. Here's an illustration of how we use 3D printing to guide serial histology. So on the left, you see another 3D printed mold that is custom fitted to the MTL region. And this mold allows for machine learning. So in the next few slides, I want to highlight some of our work that has used this ability to reconstruct histology and MRI into common space and map this information into a common atlas to answer a number of questions about anatomy as well as Alzheimer's disease pathology. So the first study was published a couple of years ago in a PNAS, and it started by just a desire to build a computational atlas of the human hippocampus and to define probabilistically the distribution of hippocampal subfields in the hippocampus. So to do this work, we had serial histology as I've shown, and in this space of this serial histology, we had labeled various anatomical subfields. We didn't trace them out, but simply labeled boundaries based on site architecture. And then these boundaries were mapped into the MRI space based on the registration. You see in the bottom left, I'm sorry. And then using that, three-dimensionally continuous segmentations were traced, as you can see on the bottom right. So this hybrid procedure involving some labeling in histology space and some labeling in MRI space gives us the best of both worlds. We're using site architecture to define boundaries, but at the same time, we're producing segmentations that are smooth and continuous in three-dimensional space. We then took these segmentations and from 9 brain donors who had histology and combined them in a space of a population-specific MRI atlas. I won't go into details on how we constructed this atlas, but it was required quite a lot of customization in order to perform group-wise registration between these very different anatomical specimens. But the image that you see is the end product of this atlas. This is the average anatomy of the hippocampus with the average distribution of hippocampal subfields within. We're currently extending this project to the entire medial temporal lobe, including extrahippocampal structures such as the entorhinal cortex in Brodmann areas 35 and 36. One of the key uses of this hippocampus atlas that we published, and particularly immunohistochemistry targeting neurofibrillary tangles and tau pathology, we have developed some digital histology analysis techniques that look at slides stained for tau and use our online platform to label validated these tangle maps in a recent paper against semi-quantitative scores and as well as just tangle counting, and found a high degree of agreement. But just want to take you here to sort of the end result of this. Once we can measure tangle density on individual histology slides, and once we can register them into MRI space, we can start to generate these beautiful three-dimensional maps of the distribution of tangle density throughout the MTL. Here are examples of 3D maps of tau density in 3 specimens. We currently have about 9 and more specific biomarkers for early AD pathology. Just another slide highlighting some very recent work where we looked at correlation between thickness and tau pathology. This is, again, using our ex vivo.
Oops. Sorry about that. Let me.
Hello, my name is Paul Yushkevich, and paper against semi-quantitative scores and as well as just tangle counting, and found a high degree of agreement. But just want to take you here to sort of the end result of this. Once we can, so to conclude, ex vivo imaging, while requiring enormous resources to process and collect the data and to analyze the data, really is unique in its ability to help us understand anatomical variation, to validate findings of in vivo literature, which is crucial because there's, when people act on these in vivo findings, if those findings are wrong, that can introduce enormous costs into the field. And so validation of in vivo findings that are going to affect the kind of biomarkers that are used in clinical trials of the future is crucial. And ex vivo imaging really allows us to do so. The future directions of this work are to continue reducing reliance on manual processing throughout the different steps with greater use of deep learning in various parts of the algorithm. We're also working on incorporating more geometrical modeling into our registration and segmentation approaches, and to expand the analysis to other pathologies, not just looking at tau, but particularly looking at TDP-43 pathology, as well as to extend to other regions of the MTL and brain regions, with one particular goal being to include the amygdala and its subnuclei. So thank you. Great.
So I'm sorry I had to fast forward through some of Paul's presentations because I realized that we're running over time. I'm actually even more apologetic that this video I don't think really does justice at all to Paul's effort, which is an incredible tour de force in fully realizing this new paradigm of ex vivo imaging that integrates MRI with histology and most importantly provides a translational link to in vivo imaging that then allows these innovations to be ultimately brought into the clinic. Now, let me close by inviting those of you who may have an interest to take a look at our lab's freely available software resources and others to which we contribute. All these that implement many of the results that I presented today. And finally, a big thank you to all of our current and former lab members and collaborators who are far too numerous to name. They deserve all the credit for everything you heard me talk about today and much, much more. We, of course, thank our sponsors too. And last but not least, my biggest thanks goes to GRASP, where the first chapters of this extraordinary narrative were written and where I took my first steps on this amazing journey in medical imaging research toward better health and better medicine for humankind. Thank you very much.
Thank you very much, Jim. That was really amazing presentation. We have a new format for the questions. As a matter of fact, we have also invited Ruzena to be panelist, but she had another meeting at 9 o'clock Pacific time. So our panelists who are going to take questions are Brandon Gonzalez, who is a robotics master student, and Kendall Quinn, who is a PhD in electrical engineering. So I'm heading this over to you guys.
Yeah, first of all, I wanted to thank you for the talk, Dr. G. That was amazing, very interesting insights in medical imaging and specifically the analysis of those images. I wanted to kind of keep the flow going as far as like kind of technical goes, and I encourage the other participants to please send in your questions, either technical or non-technical, to Dr. G, and we can kind of get to those throughout the rest of this format. But I was a little interested in you spoke a lot about medical imaging for different types for surgery planning and in some cases, as well as you know, when you do the ex vivo imaging, you can understand what has happened post-mortem. But I'm interested in knowing if you have any insights of using medical imaging for treatment and not just surgery, if that's something that happens with neurodegenerative, neurodegenerative diseases.
Yeah, for neurodegeneration is tough.
Thank you.
That's a well, both tough to pronounce and also a tough condition to have. We actually don't have a lot of therapies available for neurodegeneration. You'll see some of the other applications that are illustrated where there are more availability in terms of therapies. But you're absolutely correct. One of the exciting areas is so-called theranostics, which is this whole idea of using imaging, in particular ones that are so-called contrast enhanced, and the things for which that you're using to generate this additional contrast in your imagery, so that when they're acquired, they highlight things of interest to you, tumors or other aspects of the condition. People being extremely clever now in using that same mechanism to actually introduce the therapeutic agent. So you can both track it down in terms of being able to visualize where you're sending this contrast, and at the same time, once it's there, or where you want it to be, you can activate a therapeutic agent that's a part of this same technology. That's an area in which we actually have folk here at Penn who are working on actively. It doesn't involve as much image analysis and processing, in part because the whole idea of designing these agents is to make the contrast such that hopefully post-processing is less important, and you can rely on the acquisition setup itself to try to track these agents. But there's definitely potential partnerships there, but that's a great question in terms of ultimately that is the goal. We obviously would like to diagnose, know what's actually happening, but once you do, and if there is therapy available, be able to then direct it to where we'd like it to be.
Yeah, thank you.
Thank you, Dr. G, for the talk. So we have a question in the chat from Michael Sobrepera. So he says, I'm curious as to how clinicians respond to the new tools, excitement, and ability to understand outputs, fear the new, etc. Looking at translation, how do administrators, etc., react to that, to all of that?
Yeah, that's a super complicated situation. It goes everywhere from, like you said, convincing clinicians that this is a value add, that this is something that they ought to consider in their practical workflow, in their basic clinical practice, to all of these other situations of integrating it within standard of care that doesn't just involve actual demonstrating efficacy, but gets complicated with regulatory aspects, and so on and so forth, FDA approval and training, and so on and so forth. So the pipeline is quite extensive. It's quite complicated. Again, we have wonderful resources here at Penn, Penn Health Tech in particular. Focuses a lot on this pipeline of taking ideas from the bench and bringing it to the bedside through these various steps that oftentimes defeat the translation of great ideas into actual practice. But first up is just a basic work in terms of demonstrating utility and efficacy. And those of you who were at the celebration at the GrassFet 40 celebration, I relayed a story in which these ideas when I was first working on them, as presented to our esteemed clinical colleagues at Penn, we were shot down immediately, and our clinical colleagues thought it was a complete waste of time, and so there's these days I think our clinical colleagues are much more receptive to ideas from outside, and really appreciate the contributions that technology in particular can bring to bear on their practices. So, and I think the environment at Penn in particular has really blossomed and bloomed into one where there's such terrific interaction and engagement between the schools, and between the faculty and in this case the clinical practitioners and the researchers. I don't know if I answered the question. It was a pretty broad one, and, so I think I gave an analogously sort of like broad answer. But I can drill down further if there's something that you want to hear more about.
Yeah, no, thank you for the insight. Yeah, another question, kind of another broad one is, how have advances in hardware and computing power impacted the field of neuroimaging in recent years, and what new advances do you think could have a major impact on the field in the near future?
Yeah, that's a great question. It's sort of technology, sort of advancement of technology never stops, and it's constantly involving the clinical field, because the instrumentation that it uses is constantly evolving due to innovations that are coming. As for example, you say on the hardware side. I don't know if Brandon, if you've ever taken had the unfortunate necessity of showing up for an imaging scan at a hospital or at a clinic. If you have, the scan itself could potentially take a lot of time. Some of these are, the speed at which you take these scans can have a bunch of implications. If they're too long, they limit the sort of application. So there are patients that can't be in a scanner very long. They can't stay still for very long. Children in particular, pediatric patients have a fairly hard constraints in terms of like what you can do to them with scanning. And so there's the ever-present sort of need and interest in increasing the speed of scans, and hardware has certainly made a big difference in this. But I think that the biggest changes that have affected image acquisition per se, and certainly has been relevant to image analysis too, the analysis once you've gotten images, has not just been on the hardware side. It's really been led more on the software side. On the acquisition side, there have been some, incredible sort of speed ups due to applications of compressed sensing. So that was really hot for a while, and I think that if you were a few years older, that would have been the hot topic for you to study if you were in signal processing and so on and so forth. But that's kind of taken over. I shouldn't say taken over an interest, at least for trainees, by techniques like machine learning and deep learning, where the potential has we're only scratching the surface in terms of potential, both in terms of accelerating acquisitions and in introducing wholly new ways of doing the kinds of analysis that I presented.
Yeah, thank you. It's funny. I have gone in for a scan. I've had a MRI of my knee, and that was like 40 minutes or so. Yeah, so it was a little interesting. I was able to kind of fall asleep, and next thing I know, I was out. So it kind of worked out for me. But yeah, that's I agree. Like the time frame of capturing images and capturing scans is definitely something that could be improved upon. One of the other questions, so something that came up that kind of brought me back to my research with event cameras was kind of like this idea of dealing with 4D, in this case, it was 4D aortic representations and such. And I saw that there was like some cross sections of the valve that she was looking at. And I was interested in understanding if there were other ways to kind of take something that has these 4 dimensions, where you have the physical and as well as the changes over time, and if there were other ways to kind of break down and gather more information than just taking cross-sectional over some time period for that. I don't know if I'm making my question like clear or not.
I don't think I fully got it, but I
Okay.
think that I think I get a sense of what you're asking about. Or are you interested in sort of alternative representations of this type of data, or are
yes.
you interested in? Yeah. Okay. Well, this is the kind of stuff that I think your advisor Costas would be, would be expert in. I mean, so what's interesting is that these are just paths, right? These are paths, spatial paths parameterized by time. They're dimensional structures that we're actually all familiar with, and they're, so a lot of the stuff that you do in robotics, that's slightly more theoretical, would all apply here. They are, they're paths over time. I had talked a little bit about, these diffeomorphisms, which are, just basically, velocity fields that you integrate over time to get these displacements over time. And people have sort of, explored this idea of using these manifold structures defined on these diffeomorphisms to, try to come up with more interesting things. And as I had mentioned, it's a very active area of research. There's still a lot of people very interested in this. Admittedly, these sorts of interests wax and wane. There are trends in academic research, and I'd have to say, sort of, some of the manifold-based work in terms of these sorts of representations have been tempered a bit, again, by sort of the overwhelming or this pivot of interest now to completely data-driven sort of machine learning-based approaches. Mm-hmm. I'm a kind of two minds of this. The manifold stuff is kind of interesting because, as I mentioned, you can actually do proper math on those manifolds. You can actually compute distances, and once you can compute distances, you can do statistics. Mm-hmm. So it'd be very cool that you can do these data-driven techniques where you can, say, compute an average configuration. I'm sure you can do that as some kind of a neural net. But the flip side of it, if I were to do this on manifolds, I can actually show you the math that's actually used to derive the statistic. So I think it goes hand in hand. I think ultimately the answer to your question is the particular task for which you're interested. Yeah. And having all of these tools is really cool because depending on the question that you're interested in answering, these days there is just so much. There's just so much technology and so much math available to study questions in biology and medicine that in the past were very, very rudimentary, and now we're in this sort of like golden age where there are more things that we can throw at this stuff than we have time to do.
Sure. Sure. I'm glad you answered the question that way because it actually segued right into the last one. I think we might have enough time to answer it. I just wanted to know where you saw or what topics or what kind of growth you would see from PICSL in the next couple of years in regards to this type of analysis, in regards to the methods of manifolds or you know, skewing or moving more towards, you know, where this generation is with deep learning and such.
Yeah, so the interesting thing is that what drives the methodology, you know, I reviewed some of the work that was done at the lab 30 years ago. And the fascinating thing is that that work was motivated by a very specific question. And 30 years later, these questions are still relevant. So they're still asking the very same questions. Certainly in the kind of work that we're doing, the domain, these kinds of questions of you know, where things are, how are they different between different individuals and in different populations, how do they change over time, and, you know, all of these things are all of these questions are still completely relevant and are still the fundamental ones that drive the current work. So, you know, typically when I give these kinds of presentations, I actually try to highlight the questions more than the answers, because as you'll see over the generations, the very same question has been answered, you know, in different ways, because the tools that were innovated were different. And so people might consider the original work, you know, the original solutions based on deformable models, you know, that worked out things like, you know, regularizers and all of this kind of stuff, and then subsequently replaced by or, you know, reinterpreted using probabilistic techniques to allow, you know, not just using mechanics or constitutive equations to describe, you know, how things can potentially spatially arrange, and instead being able to encode sort of biological variability and not model it as, say, as an elastic thing, but if we could actually statistically model this, you know, use statistical models instead of biomechanical models, you know, all this stuff is each generation, and you guys are going to come up with a completely different solution to the exact same problem, and it's going to be relevant and interesting and important for, you know, the reasons for which you've innovated this new solution. So I think what we do as mentors is we sit back, you know, we present the question, we show you what's been done, what the challenges are, what the limitations are to the existing solutions, and then maybe point the direction toward new directions that may be interesting in terms of answering the exact same question, but you now doing it in your own really cool way, and hopefully it gets taken up by the community. At the end of the day, what's interesting in this field and in much of sort of what you do is that, you know, it's not just us who are asking these questions. There are literally thousands of investigators, thousands and thousands of investigators out there who have the exact same question. They're collecting their own data. They need to run their studies, and they're looking for answers. And so when you're able to present a decent answer, you have a whole audience out there who's ready to use it, and that's something that's interesting in our work, because we do have techniques that are widely used by the research community, and that in itself also then provides a source of additional innovation because of questions that follow up from the original ones.
Yeah, thank you so much for your presentation and your answers.
Great.
It's been a great insightful conversation. For sure.
Great, it's been fun.
Yeah, thank you. Gabby or Costas? Gabby, gotcha.
Sorry. Thank you so much, Costas, Jim, panelists. I really appreciate everything today. And thank you all for joining us here today. I just wanted to please turn you into, please tune in next week, Friday, October 2nd at 11 a.m. Eastern Standard Time for our first speaker in our new Grassman Robotics series, Leila Taimaka from the University of California. For information on this or upcoming events, be sure to follow us on social media or check out our website. Thank you again, everyone, and have a wonderful day.
Great. Thanks.